Hi, welcome to Chapter 3, Cellular Form and Function. We are going to go over each one of these together and figure out what they do and where they exist in a cell. So first we want to start with the outermost part, which is the plasma cell membrane. And it surrounds the cell. It defines the boundaries where the exterior boundary is, where the interior boundary is. And it's made up of proteins and lipids. We call them phospholipids because it's phosphorus and lipids, but it is protein. Now, inside of the cell is called the cytoplasm. And the cytoplasm is where all the organelles are. Okay, so all these organelles are in the cytoplasm. And it does have stored foreign particles in it as well. And then there's a fluid, which we call the ICF, okay, intracellular fluid. And the intracellular fluid is called cytosol, okay, it's called cytosol. Now, outside of the cell, we have an extracellular fluid, and we call that interstitial fluid. Now, the plasma membrane is the border of the cell, and it has both an intracellular and an extracellular face. So it has a face that faces inside the cell and a face that faces outside the cell. Its function is to actually define what the cell's boundaries are. It governs what kind of interactions are going to happen. In other words, what molecules can come in and what molecules can go out. And it can change. So those are some of the things that happens with the cell membrane. Now, again, the membrane is made up of phospholipids. That's phosphorus and lipids. We sometimes call lipids fats. So 75% of the membrane are lipids, okay? And they are amphipathic, which means molecules are arranged in a bilayer. So we see this bilayer here. So you have the hydrophilic head and the hydrophobic tail, and you have two sets of them. So it's a bilayer. Hydrophilic, philic meaning love, hydro meaning water, so it loves water. And this will face the water side of the cells or outside the cell. So in the extracellular fluid, it's facing water, and on the interface, it's facing the cytoplasm, which is mostly water. Now, towards the center of this bilayer is a hydrophobic fatty acid tail, and they avoid water. So they try to keep water from being too much inside the cell. And so these do move. They are fluid, and so they drift laterally. Now, cholesterol makes up 20% of this membrane, all right? So cholesterol makes up 20%. Without cholesterol, our cells, when they were cold, would just be rigid and nothing could go in or out. And if they were hot, everything would go in and out. So you need the cholesterol because it basically maintains your fluidity and your stability of that particular cell. Now, glycolipids make up another 5% of this membrane. So phospholipids have short carbohydrate chains on the extracellular face, okay, facing out towards the ECF. They contribute to the glycocalyx, which is a coating on the cell surface to help the cell maintain what it's pulling in. In other words, it's helping to pull things in. It also holds sugars. These sugars help the cell membrane to survive. So what is a membrane protein function? So again, something else we have in the cell membrane are proteins. One kind of protein is a receptor, so it's going to receive any chemical messages coming from the ECF. It can be an enzyme. These enzymes actually break chemicals down and terminate them so that they don't get inside the cell. Remember, this is just a messenger. It's just giving us a message. They might be channels. If they're channels, they might be open and allow certain particles or ions to move from outside the cell to inside, or vice versa. They might be a gated channel, which means they can open and close according to what ions you want to come into the cell or go out of the cell. They can just be a cell identity marker. They tell other cells what they are. And then last, they can be a CAM. With a CAM, it binds cells to other cells. So those are the different protein functions, membrane protein functions. Make sure you know those. So again, channel proteins allow hydrophilic solutes and water to pass through the membrane without any problems. We do have leak channels, which are always open. That means it leaks ions in and leaks ions out. We have gated channels. Now, this ligand gated channel respond to very specific chemical messengers, okay? The voltage gated channel only responds to electrical charges. We do have electrical charges running through our body all the time. And then you can have mechanically gated channels, and these respond to physical stress. Think about a molecule trying to get in, and it's pushing, pushing, pushing. That would be a mechanical stress. Or too much of something on the inside of the cell and it needs to get out. Same thing, pushing, pushing, pushing. So remember, these channels, these gated channels, are very crucial to both your nerve and muscle functions. Now, the glycocalyx is this coating on the outside of the plasma membrane. So it's on the external face. It's unique to everybody. Everybody's different. So these glycoproteins and glycolipids are for protection against foreign bodies, immunity to infection, making sure that tissue that is transplanted into your body is compatible with the rest of your cells, for embryonic development, fertilization, defense against cancer, and cells adhesing to one another to make tissue. So that's the glycocalyx's job. Now, also on the outside or external face of this, you find these little microvilli. Now, they sort of look like finger projections, right? So they give you 15 to 40% more of your surface area. They also are specialized for absorbing anything you need inside the cell. So cilia are hair-like projections that are outside. Again, we're still on the outside or external portion of the cell membrane. You have some non-motile cilia. That means they don't bend, they don't move materials, but they serve as antenna. They can sense when something is going on around the cell. And then many functions do depend on these non-motile cilia. The motile cilia, on the other hand, bend and move materials, either trapping it and pulling it into the cell or moving it along. So they beat like waves. Some cells have a flagella on the outside. A flagella is a functional flagellum that moves. You do find them on sperm cells. They're whip-like structures. They're few in number. They don't do a back and forth motion, but more like a corkscrew motion, and it helps to propel the sperm. Now, some cells have pseudopods on them, and that just means that their cytoplasm changes shapes and sizes to move the cells around. And these pseudopods can also capture foreign particles, bring them in to destroy them with another organelle that we'll talk about in a few minutes. So let's talk about membrane transport. Now, this is what your lab is going to be mostly about this week. So you can have a selectively permeable membrane in a cell. Selectively permeable just means it decides what can go in and out. Now, you have two types of transport. You either have passive or you have active. With passive, you do not have to use up any energy. ATP is our energy currency. And it's involved in a concentration gradient. Okay, that means it's going to move from an area of high concentration to an area of low concentration. Simple diffusion, osmosis, and facilitated diffusion all go under passive transport. With active transport, you move up or against the concentration gradient. So you're moving from an area of low concentration to an area of high concentration. The sodium-potassium pump is very important in many functions in our body, so that's one. The other one is a vesicular transport, which we'll talk about when we talk about things like the lysosome, the ribosome, some different things. And then we have this carrier-mediated mechanism. It's neither passive nor active. And what happens is that something is just helping an ion to move across the cell membrane. So it can be facilitated or it can be this sodium-potassium pump. Now, with filtration, particles are driven through the membrane by pressure. Okay, so they're actually driven through the membrane by pressure. So filtration of water and small solutes go through gaps in capillaries, capillaries being the smallest blood vessel we have. In this way, we can deliver water and nutrients, or we can get rid of waste. With simple diffusion, remember this is a passive transport, net movement of particles go from an area of high concentration to an area of low, so high to low. Substances move from areas of higher to lower concentration, and it does not require the membrane to be anything but selectively permeable. So it has to be a permeable membrane, and we move from an area of high concentration to an area of low concentration. With simple diffusion, though, there are some factors that affect this. So how quickly the diffusion happens can depend on temperature. The higher the temperature, the faster it moves. Molecular weight is another factor. If you have large molecules, they move slower. The steepness of the gradient. So how much difference is there in this concentration? If there's a lot of difference, it'll move quickly. If there's a little difference, it will move slowly. Surface area has a big part in this, in the fact that the membrane surface area, the larger the membrane surface area, the higher the rate, and again, permeability. The more permeable it is, the higher the rate. Now, osmosis is another form of diffusion. It just has to do with liquids or water. So the net flow of water is through the selective permeable membrane, and it moves from the side where water is more concentrated to where it is less concentrated. And we can also say that water moves towards areas of higher solute concentration. Now, solute are solid particles, okay? Solutes are things that we're trying to dissolve. So it can move from an area of low solute concentration to an area of high solute concentration. So water uses very specific channels called aquaporins. So let's look at these two pictures. Here's just water, and here's water with solute. Notice the water levels are exact same height, but the water is going to move where there's more solutes. And you start getting a hydrostatic pressure actually pushing down on the water and an osmotic pressure pushing away from the water. So when we talk about osmolarity, we're talking about the number of osmoles per liter of solution. So body fluids contain a mix of many chemicals, and the total osmotic concentration of all solutes is called its osmolarity. So remember, we're looking at this osmotic concentration. For blood plasma and tissue fluid and intracellular fluid, we have 300 milliosmoles. Now that's osmolarity. Now for osmolality, we're talking about how much osmolarity we have per kilogram of water. So we're thinking about a weight. So the higher the osmolarity to osmolality, then the more solute is in the concentration. A lower osmolarity to osmolality means a low solute concentration. Make sure you watch this video. It really helps to understand osmolarity and osmolality. Now, talking about tonicity, we're talking about the ability of a surrounding solution to affect the pressure in a cell. So this all depends on how many solutes are in your intracellular fluid. If you have a hypotonic solution, this causes the cell to absorb water and swell. It might even burst. The word we use for burst in anatomy and physiology is lyse. So if you have a lower solute concentration in the intercellular fluid, and distilled water is going to just rush in and it is going to burst that cell. Now with the hypertonic solution, the cell loses water, shrivels up. The word we use for shrivel in anatomy and physiology is crenate. Here you have a higher solute concentration in the ICF, and water is just being pulled out of it. With isotonic solution, nothing happens because you've got the same amount of solutes and water on each side of the cellular membrane, and it just stays its normal shape and size. So here's a picture of a hypotonic red blood cell. Here's a hypertonic red blood cell. And here's an isotonic red blood cell. You'll notice this one's very large because it's swollen up with water. You'll notice with the hypertonic, it's crenated because it is losing water quickly. And you'll notice with the isotonic, this is what a red blood cell normally looks like. So now we're to carrier-mediated transport. With carrier-mediated transport, we're actually helping to carry something in or out of the cell. So it can be very specific, like with these ligand bands, which only carry proteins, or it can happen because of saturation, where the concentration just gets so big that you got to start carrying some of the stuff out. You can't wait for the channels to open and close. So again, transport proteins are going to help you move ions in and out of the cell. So the three kinds of carrier proteins are uniport, symport, and antiport. With uniport, you can only carry one type of solute. That's it. It's very specific. So we see this in aquaporins because it only carries water across. With a symport, you can carry two or more solutes at the same time. So a sodium glucose transporter is an example of this, where you bring both sodium and glucose in at the same time or take it out of a cell at the same time. With the antiport, Now you're going to bring one ion in and push one ion out. The sodium-potassium pump does this. So sodium comes in, potassium goes out, and they keep doing that. With facilitated diffusion, the carrier moves solutes down a concentration gradient. In other words, it's going to move an ion from higher concentration to lower concentration. It does not use any ATP, and basically it's an open and closed gate is what it is. So primary active transports carry solutes up the concentration gradient. So it's going from an area of lower concentration to higher concentration. You must use ATP. The sodium-potassium pump is a good example of this. So the sodium-potassium pump, let's look at it a little closer. So basically what it's going to do is it's going to, in our picture, move sodium out of the cell and potassium in. So for every 3 sodium ions you move out, you've got to move 2 potassium in, and you've got to use an ATP to do this. So notice that the sodium is moving up a gradient and the potassium is too. So they're both moving up a gradient. So whenever it's resting, in other words, it's not moving anything, then it is negatively charged. When it isn't resting, it will become positively charged. And you should watch this video of an animation of a sodium-potassium pump. All right, let's look at vesicular transports. So vesicles are just little bubble-like enclosures of the membrane, and it carries things back and forth across the cell membrane. So endocytosis is when you're bringing things into the cell, and there are 3 types. There's phagocytosis, which means you're bringing in large particles. There's penocytosis, where you're bringing in small droplets of a molecule. And there's receptor-mediated endocytosis. And this is when particles bind to a very specific chemical receptor on the plasma membrane. And that chemical is brought into the cell. Now, the next one is exocytosis, and this takes things from inside the cell and pushes it out. And then there's transcytosis. With transcytosis, it's just you're moving materials across the cell itself. So not outside or inside, just around the cell. And these all use ATP. So the cytoskeleton is a network of filaments and cylinders, and it determines the cell shape. It supports structures within the cell. It organizes where things are inside the cell. It directs where things go from one part of the cell to another. And it's made up of 3 things: microfilaments, intermediate fibers, and microtubules. So we are going to look at all these different interior or internal organelles. They all carry out specialized metabolic tasks. So the first is the nucleus. It is the largest organelle we have in all of our cells. Every cell must have at least one nucleus, and many cells have more than one nucleus. So surrounding that nucleus is a nuclear envelope. It is a double membrane again. So just like the cell membrane, it's doubled. And it has holes in it called nuclear pores to allow nutrients to move in and out of it, as well as DNA to send messages to the cytoplasm. So the nucleus contains chromatin, which is your DNA and protein together. And the nucleolus, which is in the center of the nucleus, and it's a really dark center, makes ribosomes, and ribosomes are very important in making proteins. Then we have the endoplasmic reticulum. Now, the thing about the endoplasmic reticulum is it's continuous with the nuclear envelope. So it is surrounding the nuclear envelope. And it synthesizes, edits, and modifies proteins. So one of the things it does is synthesize. So it's going to make proteins first. Then once it makes them, it's going to help edit them. Is this the protein we wanted, or do we need a different protein? And it can also modify a protein that's already been made. We have two types of endoplasmic reticulum. We have a rough, which has the ribosomes on the outside, or we have a smooth. The smooth does not have ribosomes. Again, it's continuous with the rough ER, and it makes steroids and lipids. Remember, lipids are important for cell membrane, so that's important. And then it helps us detoxify alcohol and other drugs. If we take, let's say, too much aspirin one day, instead of our body going into an overdose, these actually help just to detoxify it. And then it does calcium storage, and this is important for our bones and our muscles. Now, the ribosomes, which sit on the outside of the rough ER, can also be found in, remember, the nucleolus, and they assemble amino acids into proteins that are specified by our DNA or our genetic code. Proteins are important to make the cell function. That's what they're for. They're to make the cell function. Then we have a Golgi complex, and this is a system of cisterna that finishes your protein synthesis. So it receives newly synthesized proteins from the rough ER, sorts them according to putting them together, packages them, and then in a vesicle sends them out to the cell. Now, they can go to and become a lysosome, or they can go to the plasma membrane and fuse to it, helping make more plasma membrane or repairing damage, or they become secretory vesicles. In other words, they take protein out to be used later in the cell. So here's a Golgi complex. Here's their vesicles. Here's how you see it on a microscope slide. Now, a lysosome has a bunch of enzymes in it. And those enzymes are used to digest or destruct materials that are either a foreign body that we don't want in our cell, or it can be a damaged organelle that needs to go, so they digest it. Or they might just digest a protein, a nucleic acid, a carbohydrate, a phospholipid, some nutrient that comes into our cell. They digest it so we can use it. So again, therefore digestion. Now they do autolysis. That means if it's time for a cell to not exist anymore, they will actually burst it. Now peroxisomes resemble lysosomes, but they contain a very different enzyme. These neutralize free radicals that are in our bodies. They also detoxify alcohol and other drugs, and a variety of bloodborne toxins. So we get a lot of bloodborne toxins coming through our body, and we need these peroxisomes to get rid of them. They are very abundant in both our liver and our kidney. Now, the mitochondria are what make those energy currency of ATP, and so they, again, are double membrane. They have their own cristae, which are folds, and the spaces between the folds are called the matrix. They are one of the few organelles that have their own DNA. It's called mitochondrial DNA. Now, the thing about mitochondrial DNA is that it's only from your maternal side. So, only your mother gives you DNA that sits in your mitochondria. And these are called the powerhouse because they make the ATP. Centrioles are found in animal cells, and they are just microtubules arranged in groups of 9s. We find them usually just floating around in the cytoplasm until it is time to do mitosis, which means divide the cell. At that point, they move to opposite ends of the cell and they start helping with the function of mitosis. They are also the base of the cilia that we find on the outside of the cell membrane, or a flagella that we would find on the outside of a sperm. Now, there are two types of inclusions. There are stored cellular products, such as glycogen, which is a sugar, granules, pigments, which give us our skin coloring, fat droplets, which store energy, or they're foreign bodies, such as viruses, bacteria, dust particles, debris, things that we want to get rid of. They never are enclosed in a unit member, and they are not essential for survival. So you do not have to have inclusions in your cell. Between each cell, we have a very tight junction. Now, these tight junctions are there to seal off intercellular spaces so that substances can't pass between cells, all right? They are like a lock and key, so they are called tight junctions. There are desmosomes. These are, again, ways to hold two cells in a proximity to each other, but not to hold them so tight that materials can't get between them. They have, again, a J-shaped hook. So hook, hook, all right? So their transmembrane proteins help these cells just join together so that things can move between them. So that's called a desmosome. Your skin does this. Now, gap junctions, on the other hand, are ring-like, and they allow ions, nutrients, and other small solutes to pass between cells. They are located in your cardiac and smooth muscle, as well as embryonic tissue. Now, stem cells can be of two types. They can be embryonic stem cells, which means they're either totipotent or pluripotent. Totipotent meaning that they can become any cell. Pluripotent means they can become many types of cells. So, again, you've got these two types of embryonic stem cells. Now, adult stem cells can be multipotent, which means they're able to develop into two or more different types of cells, or unipotent, where they can only become one type of cell. If you have any questions about cells, please let me know, and I'll be glad to share with you.